This video lecture is over Chapter 6, Genetic Analysis and Mapping in Bacteria and Bacteriophage. In the previous chapter, we spoke about how genetic recombination can occur in eukaryotes during meiosis through the production of haploid gametes that then recombine during sexual fertilization and sexual reproduction, as well as crossing over that occurs during that process. Today we're going to talk about prokaryotic cells and how they can engage in genetic recombination. Now, prokaryotes primarily reproduce through binary fission, which is a mitotic division to where you have one cell and then you have two cells, and that's their division. But there are ways in which we can have genetic recombination. Now, when we talk about bacteria and prokaryotes, often we find ourselves talking about the adaptation hypothesis that says one of the ways in which bacteria can evolve and have genetic recombination is through interactions with viruses, bacterial viruses, namely bacteriophage. And that this interaction between the two becomes essential for the bacterium's ability to acquire immunity to that particular phage. So what this means in short in the adaptation hypothesis is the more exposure a bacteria has to phage, the more likely this is to result in resistance in the bacteria. And the question comes, where does this come from? Well, this resistance can come from spontaneous mutation that occurs either in the presence or absence of a phage, which just through DNA replication, a mutation error is made, or a replication error is made that results in perhaps immunity. Okay, that's the primary source of genetic variation in bacteria. Bacteria primarily are evolving just through replication and those mutations that occur. The life cycle of a bacteria is such that they have three growth phases. For those of you that have taken microbiology, this should be review. For others, maybe it got covered in 112, maybe not. So just as a short primer and review, let's talk about what happens to bacteria during their growth phase. So in the beginning of the growth phase of bacteria, let's say it's just replicated, you plate it on a media, we enter what's called the lag phase, in which it takes a little bit of time for that bacteria to start growing and then replicating. Shortly after the lag phase, we enter this exponential log phase, in which the bacteria are exponentially dividing. And at some point, we're going to hit a carrying capacity in the media, and we'll hit this stationary phase where we're just not able to create more bacteria because there's not enough essential nutrients for those bacteria to survive. Now, how do we get genetic information from one bacteria to another? How can we have this genetic recombination? Well, there's three ways. This can occur through conjugation, through transformation, and finally through transduction. We'll begin with conjugation. Conjugation is where genetic information is passed from one bacterium to another. And when it's passed, it then recombines with that second bacterium's DNA, and we have genes that are passed on. Now, this occurs in bacteria through a specialized fragment of DNA known as an F factor. So just as a primer and reminder, bacteria, they have, again, two copies of DNA, but those DNA in prokaryotes are circular, right? They're called a plasmid. Okay, they're circular. The F factor is a small plasmid in and of itself. It's separate from the chromosome of the bacteria. And in some bacteria that have these S factors, the F factor stands for fertility, and the genes that are found on this F factor basically encode for the proteins necessary to create the sex pilus, which is the conjugating structure that allows this bacterium to attach to another bacteria, perforate its cellular membrane, and then it also contains enzymes that nick the fertility factor and allow it to synthesize a new strand of DNA and pass that on to the conjugating bacteria. So when we go through an entire cycle of conjugation, if we have what we call an F plus bacterium, that means it has the fertility factor. Let's say it conjugates with an F minus bacteria, one that does not have the fertility factor. At the end of recombination, both cells will now contain an F factor. Okay? The amount of genetic recombination is actually very low in this type of conjugation, an F plus with an F minus, because we really didn't add too many extra genes. No genes in this chromosome, just the fertility factor itself. So again, this is just going through the process. The F plus cell serves as the donor DNA. It contains that fertility factor. It creates the sex pilus, okay, that then serves as the conduit by which the bacteria shares, and then that F factor gets transferred and copied to the F minus, and we end up with an F plus cell. Now, sometimes we have what's called high frequency recombination, in which the bacteria strain takes its F factor, that plasmid, and it actually inserts it into its own DNA. In this case, we now contain all of the genes necessary to create the sex pilus, to nick the DNA, to ligate. All of those protein, or all those genes exist now within our DNA. And when we do this, we can now have a very high rate of recombination. Okay? And the way we know that recombination is occurring in a high-frequency recombinant bacterium is through what we call interrupted mating. So there was experiments done in which we had these high-frequency bacterium, and they were allowed to conjugate for relative periods of time and kind of stopped. And what they found is that, the more time that went on, the more genes were actually transferred to the other bacteria. And not only that, but they were transferred in gene order. And so the distance between these genes dictated, or you could predict, how long it would take to pass all those genes. What this looks like, if we look at gene transfer, let's say we have a series of HFR strains. So here's our strains. And we see this is their order of transfer. So what happens is somewhere in the DNA molecule is our F plasmid. In each strain, maybe that F plasmid's in a different spot. Depending on which, whether we're on the outer ring or inner ring, and which, where our origin site is located at, that's where the fertility factor is in, we'll start passing on those genes in a linear fashion. So for this first one, for the HFR strain, here is our origin. This is on the outside strain. So we'd start with passing first. The first one that'll be passed is going to be the THR, leucine, our AZI, TON, PRO, LAC, GAL, and then Thi. That would be its order. This one, maybe we're going the opposite direction, so we'd have leucine, Thi, GAL, LAC, PRO, TON, and AZI. So on and so forth. It's a really easy way to kind of do this. So let's look at an example. This is a problem that you could expect to see on your first exam. Something like this. It says, okay, here's our strains. This is the order. What we want to know is, what is the actual gene order of that chromosome? And the way we solve this is pretty simple. We just are going to write these out. And remember in our heads that there are circles. So for the first one, I'm going to just write out our gene order. So our gene order is T, C, H, R, O. And we could be going forwards or backwards, depending on where that origin is. So for a second one, what I'm going to do is I'm going to start somewhere where I have overlap. So H, then it goes R, then it goes O, then it goes M, then it goes B. Okay, for the next one, we start with M. So I have an M, and then it says the next one is going to be O. Now, I don't want to write O here because obviously I'm going in the opposite direction. So I'm going to go O, R, H, C. Okay, for strain 4, again, starting with M. Then it goes B, A, K, and then I'm running out of space, but then it goes T. And on the last one, I have C. So I'm going to keep that in the line, T, K, A, B. What you see is that T, K, A, B is here. And so we have a circle. So if I'm going to draw out what's the gene order, I'm going to draw a circle because in bacteria, right, our DNA is on a circle. And I'm going to just start writing. I'm going to start from T. So T, and I go C, H, R, O, M, B. I'm going A, K, and then I should be again at T, C, and I just keep going around like that. That's the gene order. That's the order at which we had it. And for each of these strains, I could show where the origin is if I want, but I'm not going to ask you guys on the exam. I'll just ask you to kind of do this. Okay, so we talked about how we can do gene mapping with a high-frequency recombination. Let's look at what's actually going on in this high-frequency recombinant. Okay, so we start first, we have an F plus cell. At some point in time, that F factor gets incorporated into our genome. Okay, so here it is. Now we have this origin site. Now this HFR cell, let's say it conjugates with an F minus. Okay, it's going to nick at the origin, and then it's going to start passing, moving, away from the origin. So we're going to start getting this big A, big B, big C, depending on how long they're together, right? At some point, these bacteria are going to get interrupted. They're going to separate, and so we'll have a specific amount of DNA that gets passed. So let's just say that it's a short period of time and we only pass A and B. Okay, so we try and transfer across. We then have replication occurs on both strands, so we fill back in what we gave. We also have replication right here. And we then end. One thing that should be noticed is we never pass the F factor. Unless we had a really long time, which very rarely happens, we would never get that F factor being passed. All that got passed was this A and B gene that was on the genome of our HFR. Okay, now at this point, this is not incorporated into the recipient's DNA. Okay, what happens is we create this is what's known as a heteroduplex. Okay, and at some point in time, we can then have, when we go through replication the next time, we can have a crossover that occurs here, and we can have some of these being incorporated into the genome of the recipient. Okay, we can see this happening to some extent in other cases where we'd have like the Mayer-zygote. So end result, though, of what we have is we still have our HFR. The F minus cell is still an F minus, but we had a high degree of recombination because we actually got genes from the donor cell's genome to be passed into it. So recombination is high, but we don't get the fertility. This cell itself can't conjugate with another cell because it doesn't have a fertility factor. In some cases, once we have an HFR cell, what can happen sometimes is we can create a loop in which we have enzymes come in to try to fix this loop, and they excise out the fertility factor back out of the genome. But in the process of doing that, we perhaps excise out some genes as well. And then these genes are contained on the F plasmid. We call this an F prime cell. Okay, so with F prime, conjugation again can occur with an F minus. And in this case, we'll pass not only the fertility factor along, but we can also pass on those additional genes. Those additional genes are kept in in this heteroduplex, in this plasmid outside. We call it a merozygote. And there can be recombination that can occur potentially between the merozygote and the recipient cell's DNA, but it's still going to produce those A and E genes. Like the proteins will be produced because they'll be expressed. So what we end up at the end of this is 2 F prime cells. So the F minus becomes F prime. Relative to the amount of recombination occurs, more recombination occurs in an F prime conjugation than just an F plus, but it's less recombination than if it had been an HFR, because there's only a small number of genes that are found attached to that F prime. Okay, I've used a couple of terms. I probably should have defined them a little bit earlier, but let's just define these now. So plasmids are composed of double-stranded, closed-circled DNA. Okay, sometimes we can have multiple copies of these in the cytoplasm. They may contain one or more genes, and they are going to replicate independent of the bacterial's chromosome. So, for instance, the fertility factor was a plasmid, right? So we can have fertility factors. Those confer the ability to conjugate. We can have what we call R plasmids. These generally confer antibiotic resistance to bacteria. We can have Col1 plasmids. These encode for proteins called colicins. These act as kind of poisons for neighboring bacteria, so it produces toxins that don't allow other bacteria to grow near them. These are some common plasmids found in bacteria. Okay, so that covers conjugation. Let's talk about our next way, but we can have genetic recombination, in which we're kind of shuffling genes around or acquiring new genes in prokaryotes. In transformation, we have small pieces of extracellular DNA that are taken up by living bacterial cells and integrated into their chromosome. So here we have what we call a competent bacterium. Competent bacterium have receptor sites on their plasma membranes that allow them to bind double-stranded DNA found in the environment and bring it into the cell. So we have these extracellular DNA, it's lying around, the competent bacteria binds to it, and then it brings it into the cell itself. We're going to we're going to become single-stranded. One is going to end up degrading. The other strand is going to bind to the complement sites in the bacterial genome. So let's say this is, loci A, loci B, and here in its genome is loci A, loci B. It binds, and we form, again, what we call a heteroduplex. Okay? Because it gets transformed in, so we excise out the part of the genome that corresponds to that section. So now we have different. And what's interesting to remember about prokaryotes is that at all their alleles, they generally have the exact same. They are homozygous for all these alleles, right? Because we don't have sexual reproduction; it's just dividing itself and making replicants. So these orange strands should be exactly the same. But now we have some difference. So here, at this point in time, we could actually have a heterozygous individual at these loci. But what happens is when this cell divides, after one round of division, so this inside chromosome is going to replicate itself, and so we'll have copies. This would be called a non-transformed cell. It doesn't have any of the new alleles. But the transformed cell, when it replicates itself, it makes a copy that has the complements to it. So now it's all homozygous, but it's transformed. We have those new alleles that we previously didn't have, right? Have been are now found in the transformed cell. So that's transformation. The last way that we can have genetic recombination in prokaryotes is what we call transduction. This is virus mediated. So viruses that attack bacteria are called bacteriophages, And what they do is they infect bacteria by injecting their DNA into the bacteria, and then their DNA serves as templates not only for it's the template material for the bacteria to produce all the parts it needs to make more viruses. So it produces progeny of the phage particles. Viruses can't replicate on their own. They hijack the cell's cellular mechanisms to copy DNA and make proteins. Okay. These then get released from the host when it's lysed and it's destroyed. So if we look at the life cycle of a virus, so our virus infects, it doesn't get absorbed, it injects its DNA in, that DNA then degrades the host's DNA, right? And it assembles itself into it to some extent so that it can then bind in. Then as we replicate, we start creating all of the genes on that phage DNA, produce the proteins needed to make the capsids of the new viral thing. And once these mature, then they'll lyse and be released to infect another one. So we have these kind of two stages. We have lysengy and the lytic stage of viral infection. During lysengy, the phage DNA integrates itself into the bacterial chromosome, and it's replicated along with the chromosome, and it's passed on to daughter cells. So we can kind of think of lysengy as when a virus is dormant. It's put its DNA into the chromosome, into the bacteria's chromosome, but it's not making more viruses. And as that bacteria divides, it then replicates that viral DNA and passes it on to all the daughter cells, and it then stays with those new daughter cells. So when we have this prophage, we're called lysogenic. We can grow, divide stably until that viral reproduction is induced. At that point, we move from lysengy and we move into what we call the lytic stage, which is what we see here. We start producing viruses and they lyse themselves out. Viruses can be utilized for bacterial genetic recombination. So there's two types that we can have. We can have what we call generalized transduction and specialized transduction. In generalized transduction, the bacterial DNA is what is packaged into the viral capsids. So we've gone through lysengy, we've incorporated the phage DNA into it, but when we go into the lytic cycle, instead of inserting more of the phage DNA into the viruses, some of them are defective and they get bacterial DNA packaged into it, right? These get packaged in. Now, when this virus infects another bacteria, instead of injecting phage DNA, it's going to inject viral DNA, which then integrates itself into the replicated chromosome or the recipient chromosome, and now we've had genetic recombination. That's generalized. Okay, we have a large number of genes that get passed this way when we use generalized transduction because we packaged, a large portion of the recipient. In specialized transduction, what happens now is we package both a small amount of the bacteria's chromosome goes into the phage, but also the phage DNA. So that means the resulting virus has both phage and bacterial DNA in it. And what this does is that when it infects another bacteria, not only will we have that recombination, but we'll also have the phage DNA, which allows for the production of more bacteria. And so generalized transduction and specialized transduction kind of result into the same thing in the sense that we get genetic recombination, but the outcomes are different because in specialized transduction, we now have the ability to create, or that cell would have the ability to go into the lytic cycle once again and create more viral or more bacteriophage that could then infect additional. And we've tried to utilize these in different ways in nuclear medicine. So in generalized transduction, we've looked at ways of how can we create bacteriophage that contain perhaps the genes that a particular person is lacking and then could we infect that? Could we give an injection of that into a person, into the area where those cells don't contain that, and hope that the bacteria or the phage then gives the donor cells in people, that it would then be able to give those genes that are lacking. So let's take an example. There's a form of blindness that occurs. It's genetic, in which the rods don't fully form because there's a mutation in the proteins that form our rods in the eye. And so they've looked at using bacteriophage and putting in working copies of that gene, and then inserting those into the eye, that bacteriophage into the eye, to then have it attack those cells and give functional copies to those cells that then when they divide, they would be able to restore vision. And it's actually been done in humans, seems to work. The Holy Grail is getting specialized transduction to work, to where not only are you giving those injecting those viruses with working genes, but they also have viral DNA to be able to replicate themselves, because then you could administer a single dose and it continues to administer the cell, or administer that dose internally as those viruses replicate. The problem with specialized transduction is you have to, during the lytic stage, those cells get ruptured and they died. So you have to figure out a way of how could you replicate it, but not cause lots of cellular death at the same time.